Welcome, everybody. Thank you for joining us this evening for the second in our series of talks for patients about RA and RA research in acknowledgement of Rheumatic Disease Awareness Month. This is our second of three talks. We're excited that you're here this evening. Our talk this evening is Precision Medicine in RA. What can your joint tissue reveal, and can it guide treatment? Share that this presentation is a collaboration of the Inflammatory Arthritis Center of Hospital for Special Surgery, the Integrative Rheumatology and Orthopedic Center, and the Department of Social Work Programs, Inflammatory Arthritis Support and Education Programs. I'm Adina Batterman. I'm the Senior Manager of Inflammatory Arthritis Support and Education Programs in our Department of Social Work Programs. And our speaker, our esteemed speaker this evening, is Dr. Melanie Smith. Dr. Smith is the director of the Inflammatory Arthritis Center and is an attending physician in the Division of Rheumatology at HSS and an assistant professor of medicine at Weill Cornell Medicine. She specializes in the treatment of patients with inflammatory arthritis, and her research focuses on how cells normally found in the joint lining react to and regulate the influx of immune cells seen in inflammatory arthritis. Her goal is to improve our understanding of synovial biology, to identify novel therapeutic targets for the treatment of inflammatory arthritis. We're excited to start this presentation and learn more about how joint tissue may one day guide treatment. Thanks so much, Dr. Smith.
Great, thank you so much. That was a very lovely welcome. So just to give you a quick overview of what we're going to be talking about, I'm going to start with an introduction to precision medicine itself, what it is, and give an example of how it is currently in use in oncology or cancer treatment. And then we'll go into how it might be applied in rheumatology. We're not quite there, but there's some really interesting work being done currently that I think will help get us towards applying precision medicine to rheumatology. And then I'm going to highlight a little bit of the current research being done, both at HSS and also around the world, with this goal in mind. So to get started, what is precision medicine? So precision medicine customizes treatment to individual patients based on disease characteristics. So what might that look like? So in standardized treatment, all patients with a disease with the same diagnosis get the same treatment or the same treatment algorithm. In comparison, in precision medicine, patients with the same diagnosis may be divided into subgroups based on some set of characteristics and treated differently based on those distinct features of their disease. So how do we differentiate these groups? Well, there are a number of different ways that this is done in different diseases and with different precision medicine approaches. But some common ways to do this are to look at molecular characteristics of the disease tissue itself, using pathology or analysis of the tissue. There may be a marker in the blood that differentiates people with the same disease. And then there also could be genetic differences between patients with the same disease. All of these different things could be used to split patients with the same diagnosis into disease subgroups that could respond to specific medications. So why don't we do this for everyone, though? Sounds kind of nice, right? You have like the patient with the problem, you track exactly what is wrong in their disease, and then you put a specific treatment together with that patient for ideally the best outcome. But this is hard because in order to know how to separate these groups of patients with the same diagnosis, you oftentimes need to know molecular characteristics of the disease. So you have to have a good understanding of the disease from research. You have to be able to test for these differences. So there have to be approved diagnostic tests to say that one person's disease is different than another person's disease with the same diagnosis. And then you have to have a medication that works differently in the specific genetic abnormality or specific blood abnormality that is seen in these patients. So, what is the goal here? So, really, the goal is to find the right treatment for each patient to get control of their disease quickly. The goals are to have faster symptom control by having a more targeted approach and to start the right treatment early. And with faster symptom control, there can be less organ and tissue damage from uncontrolled disease. And with minimization of exposure to medications that may be less effective, there may be fewer side effects from medications. And in some cases, where medications actually target a specific disease abnormality, there can be less off-target or whole body side effects. So this is the goal. And let me just give you one example of sort of how this works in oncology or cancer care. So for example, let's say this patient has a lung cancer. The doctors will then take a biopsy of this, and then they will take that piece of their tumor and they'll do two different things with it. One, they'll look at it under the microscope to say what type of lung cancer that is. And they will also analyze the tumor's genetics to determine if there is either one genetic mutation versus a different genetic mutation. Because depending on which one the patient has, that may mean they get either treatment A with this mutation A here, or treatment B with mutation B. In the case of some forms of lung cancer, this treatment specifically targets the protein that's the result of this genetic mutation. So this is a very somewhat simplified example, but you have to, again, be able to do a number of different things. You have to have the molecular understanding of the disease to be able to test to find differences in the disease, and then you have to have medications that are given differentially between these two disease groups. So the lesson here is that when we understand disease at a molecular level, we can create more effective and personalized treatments. So there's sort of two different parts of this that you can do this sort of the pairing with medications two different ways. One is, as I've mentioned, you can sometimes develop new medications that are targeting specific forms of the disease or a mutation only present in a subpopulation of patients with the disease. Or you can also match individual patients with medications that exist that are likely to be most effective. So how does this relate to rheumatoid arthritis, right? We said this was going to be a talk about precision medicine, rheumatoid arthritis. Clinically, there are multiple challenges that as a clinician, I believe could be addressed by a precision medicine type approach. One big issue in rheumatoid arthritis is that we are not able to match the patient with their ideal medication for them every single time with the first time. Sometimes we have to try multiple different medications to find really the right medication or combination of medicines for a patient. And because our medicines are not fast acting, patients often need to be on a medication for about 3 months to determine if it is effective or not. So during that time of waiting for a number of months to see if a medication is effective, it can really end up taking sometimes many months or even sometimes years to find the best medication combination for a patient. And 30 to 50% of patients do not have a good enough response to initial medications. And as we do this, these medication trials, sometimes patients have ongoing active joint inflammation that can sometimes cause irreversible joint damage. So we think we can do better. One of the good things about rheumatoid arthritis, and where we're really lucky, is that we have many FDA-approved medications to treat the disease. This slide is really just to remind you that there are many medications, starting from the conventional DMARDs, usually where we start treatment for rheumatoid arthritis. We then may add a TNF inhibitor. This is sort of our biggest class of medications with 5 different TNF inhibitors. And then we have our non-TNF inhibitor biologics and our targeted synthetic DMARDs or JAK inhibitors. So we have a lot of options. And really, one of the questions now is how can we do a better job of matching individual patients with specific medications? Okay, so we do this a little bit already. So I'm going to show you next some of the factors that we use to figure out what medication may be best for a patient. This is by no means exhaustive and does not even begin to take into consideration a lot of the patient preferences and other medical problems a patient may have. But these are some of the factors with specifically their rheumatoid arthritis that we think about as we're recommending medications to a patient. So one is RA-associated autoantibodies, rheumatoid factor or RF, and CCP, which you may be familiar with. We also sometimes look at the markers of generalized inflammation in the body, the ESR and the CRP. We oftentimes will look at hand or foot x-rays to look for evidence of bone erosions or damage to the bones from swollen joints. And then some patients have either lung or cardiovascular system involvement with the rheumatoid arthritis. This is an important piece of information as we choose therapies. What you don't see here is any joint-specific information. So what I'm going to tell you about now is the work that we are doing, we being the larger rheumatoid arthritis research community, to try to better understand what is going on specifically in the joint tissue of rheumatoid arthritis patients and what that can tell us. So just as a reminder to get everyone on the same page, the synovium is the primary site of inflammation in rheumatoid arthritis. So here we have a number of swollen joints. If you look at a swollen joint in rheumatoid arthritis, the real site of most of the inflammation is in the synovium. The synovium is the joint lining, shown here in RA and up here in the healthy synovium. And you can see that in the healthy synovium, it is only a few cells thick. It is extremely thin, but is responsible for making the synovial fluid, which bathes the joint and also lubricates the joint so that the cartilage on both ends of the bone can slide very smoothly past one another. In rheumatoid arthritis, the synovium is really expanded and there's lots of inflammatory cells there. And that inflammation can result in damage to the adjacent bone or cartilage. Excuse me one second. So research has really been increasingly interested in studying the synovium to learn if this could be really one way to look at molecular differences in RA. Somewhat similar to what the oncologists are doing, looking at the molecular differences in the tumor in patients with cancer. So what do we hope to be able to do here? Again, the lesson from oncology is that when we understand the disease at a molecular level, that's when we can really create more effective personalized treatments. It can also help us figure out which medication may go with which patient. So our goals in studying the joint tissue are to understand the disease, what types of cells are present at disease onset, what immune pathways are activated, and how is the joint affected by treatment? Are specific types of synovial cells affected by different treatments? And are there synovial factors that predict response to specific medications? And then, of course, as we are learning all these things about what cells are there, how they're activated, who they're interacting with in the joint, we're also thinking, can we develop any new, better treatments that we could use to treat patients with even specific forms of rheumatoid arthritis? So the first step was really to understand what types of cells are in the joint lining and how do they interact with one another. So here I'm showing you a histology image of an RA patient synovium taken here at HSS by our wonderful musculoskeletal pathologist, Dr. Edward DiCarlo. And you don't need to know exactly what's going on here, but I'm going to walk you through it. So each of these dark little circles, the darkest ones you can see, represents a cell. And you can see that the circles are a little bit different types of shades of darkness. They're a little bit different sizes and shapes. And that really tells us what type of cell it is. There are also more molecular ways to do that, but this is one way in which we get an overall sense of what's going on in the tissue. And what we can see is that in addition to some blood vessels here with red blood cells in the middle, we can have a number of different types of cells present. We can have myeloid cells here that I'm going to show in future cartoons in green, lymphoid cells, which are these ones with a more dark nucleus that are really tiny. Those are lymphoid cells, and they often form little groups or like we call aggregates, or little balls within the tissue. And then we have fibroblasts, which are structural cells. So these are the cells that are there even in the healthy joint. They make the synovial fluid. They give the joint integrity and structure. And in rheumatoid arthritis, they tend to expand in number and multiply so that there are more of them than there normally would be. And what researchers have discovered is that between patients, there's actually a lot of variability in terms of the relative amounts of these cells. So here, I'm going to show a few more of these histology images along with some cartoons to show you what's going on. So in RA patients, one possible scenario is that when we look at the joint tissue, the synovium, we can see that there's tons of these lymphoid cells. And if we look at the relative proportion of cells within the tissue, shown here with this pie chart, the majority are these lymphoid cells, with a minority of either myeloid cells, fibroblasts, or other types of cells. Another pattern we see is patients with more myeloid cells, where again, more than half of these cells are myeloid type cells, and they tend to be more spread out across the whole tissue. You can kind of see these dots are arrayed everywhere. And then there's a last subset, which is the low inflammatory or fibroid, because it's full of fibroblasts. And this one tends to have fewer of the myeloid or lymphoid cells, but lots of these fibroblasts, really creating an abnormal number of them, and really too much of all the structural types of proteins that they normally make. So this might seem sort of somewhat simple, but it's really exciting because what we have found is basically a cell-based molecular difference between patients, all of whom have rheumatoid arthritis. And really, this is one stage of precision medicine where we are now identifying differences between patients. And we've now had multiple groups study this, and all of them have found similar types of patterns where either you can have lots of these lymphoid cells or myeloid or less of the myeloid and lymphoid overall, and really just the structural cells. And we can see this both in patients with very early disease. They've had it for less than 6 months, or even if they've never been on treatment and have only been recently diagnosed. And we can also see this in patients on treatment. They maybe are having a flare of their disease, or even patients with more difficult to treat who have been on multiple therapies and are having difficulty finding that right treatment. We can see these same patterns. And I just want to show you some evidence of our scientific papers that come out of this work. But these are 3 papers showing basically the same thing I've just shown you there. There is differences in the cellular makeup between patients with rheumatoid arthritis at all stages of disease. This first paper, the identification of 3 rheumatoid arthritis disease subtypes by machine learning integration of synovial histologic features and RNA sequencing data. A very long title, but saying that they found different groups of RA patients, right? And this is work done primarily by Dana Orange, who's one of our rheumatologists at HSS and a really fantastic basic scientist. This next paper is from colleagues in the UK and molecular portraits of early rheumatoid arthritis. Here they are really focusing on those patients who had yet to be treated. And they found that even in that group of patients, they had these 3 different types of cellular makeup of their joint tissue. And then finally, there's a paper which many of the people at HSS contributed to with a very large consortium or groups of researchers from multiple sites called the Deconstruction of Rheumatoid Arthritis Synovium Defines Inflammatory Subtypes. So there's a lot of detail in each one of these papers, but the real fundamental message of all of them is that we can take a patient with a diagnosis of rheumatoid arthritis with a swollen joint, and we can look at their tissue and we're going to find different things. And so now what's happening is researchers are asking if these tissue differences can help us predict treatment response to specific medications. And so I'm going to give you one sort of example of how a study like this might look. So you can start with a patient or a group of patients. Usually we need many, many patients to be able to say anything more definite about a research finding like this, especially when there's a lot of differences between patients. You usually need a larger number. And we take patients all at the same sort of treatment stage. So let's say patients all having tried one medication, but not having a good enough response. And so they're starting on their second medication. And let's say they're all going to get treatment A. So we're taking these patients, they're all starting on treatment A. We do a biopsy of their synovium, and we determine whether or not that tissue has more lymphoid cells or myeloid cells, or neither of those, but mostly just the fibroblasts that make up the joint structure. We give them treatment A, follow them forward for at least 3 months to see if they're going to have a response or not. And then we see, did the patients with these different types of joint tissue have the same response? And one possibility we could find is that patients with these two subtypes had a good response, but the last group did not have as good of a response. And so those are the types of studies that are currently being done to try to tease this apart. And here are a few different papers where researchers, and these are really all from our colleagues in the UK, where they're using this data together with treatment response to specific medications to determine whether or not they can figure out basically a biomarker or a specific test that could be used to determine if a patient is going to respond to a specific medication. So each of these patients has basically a hint or some signal that yes, there's differences in treatment response, but they're not black and white, and there's no one specific test yet that is able to say patient is going to respond to treatments A, B, or C. So these results are not quite yet ready to be translated into regular clinical care, but that is where we are heading. I think it is also worthwhile to remember how long this sort of journey in research takes. So for TNF inhibitors, the most common biologic used in rheumatoid arthritis, the molecule of TNF was actually discovered in 1975. And it wasn't until more than 20 years later, in 1998, that the first TNF inhibitor was approved for use. So it takes years and years of doing trials, studies, doing molecular characterization of patient data from those patient blood samples or tissue samples from those studies to understand what's going on and basically having enough information to say we should change how we provide treatment to patients with rheumatoid arthritis. So currently, as I mentioned, multiple clinical trials are ongoing to understand how molecular features of the synovium relate to treatment response to specific medications that have already been approved. So most of the studies here aren't looking necessarily at a novel therapeutic. They're saying we have this cornucopia of medications. How can we better bring together the right medication for the right patient? So next, I'm going to go through a little bit what it takes to do one of these trials. I think it's a really important part of research and important part of understanding how we get to being able to actually use precision medicine in the clinic. So I think the one take-home point from this slide and this sort of series of slides is that research is really a team endeavor. It's part of the reason I like it. I think it's really fun to work with my colleagues to try to really figure out a problem, to design the best way to answer a question about either a molecular thing that is going on in the joint or a larger question of how patients respond to something. So the first thing that has to happen is doctors have to come up with some hypothesis or an idea for a study. They could say, we think that patients with the fibroid or that low inflammatory fibroblast-rich synovium may respond better to treatment Z. The first thing that has to happen is they have to work with research managers to help design a protocol. They then have to apply for funding. This is crucial. Without any of these stages, it wasn't happen, but the funding really has to happen before the rest of this can. So this usually comes from the NIH, the National Institutes of Health, foundations, such as multiple rheumatoid arthritis specific foundations that fund a lot of the research that happens. And then also pharmaceutical companies do spend a lot of their money on research to get new therapeutics to the market. Next, once there is funding and there is a plan for a study, there has to be regulatory approval. This is to ensure that everything is done safely to protect the patients who will be enrolled, And this is a really important part of any study that involves human research. And then once this has been signed off on, then the study can actually get going and enrolling their first patient, which is always a very exciting time in a study. And this impacts and takes huge numbers of people. One, of course, the patients. We're always very grateful to our patients who participate in research. Oftentimes, in order to get enough numbers of patients, we often work with colleagues at other sites, other universities, um, sometimes internationally, sometimes nationally, to be able to have enough patients to be able to end up being able to conclude something from our data. And this involves research coordinators, research nurses. Oftentimes, for rheumatoid arthritis studies, we need a radiologist to look at the imaging. We need pathologists to look at those histology of the synovium. And we need laboratory technicians whose role is to really process samples that come out of patients, like blood samples or the synovial tissue samples, and store those safely for future analysis. We usually wait until we have all the samples from all the patients before we go on and analyze them. And sometimes for many studies, patients don't have to just come once, right? Patient is enrolled at one time and then they take a medication or something happens and then they are reassessed at a follow-up time to see if anything has changed in terms of their disease activity or disease characteristics. Once all the data has been collected, then there is data and sample analysis. Nowadays, that tends to involve a lot of understanding from like a study with synovial tissue samples. We're doing a lot of sequencing of RNA. We're doing a lot of characterization of specific tissue markers. We are also looking at specific blood markers. And oftentimes thinking about if there are other ways we can stratify patients to be able to see different signals in the data, like looking specifically at patients with the CCP or RF positive versus those that don't have that. And then once we have analyzed all the data and all the samples, then we have made our conclusion and we can say that yes, patients with the fibroblast synovium, they did respond very well to treatment Z. And then we go and we have to disseminate that information, right? We have to tell everybody about it. So that usually involves going to conferences to tell all of our colleagues, all of the rheumatologists at all the other places in the world saying like, hey, we found this thing. And then we also usually publish it in journal articles. So those are all really essential ways that we communicate what we found and also do things like this, where we also communicate what we found not only to the patients who are enrolled in the trial, but patients more generally, saying that we are making real steps forward in understanding rheumatoid arthritis and treatment options. So one of the groups that is doing this sort of study, both here at HSS and internationally, is the Accelerating Medicines Partnership Autoimmune and Immune-Mediated Disease Program. This is a huge consortium and has been one of the places where we've done this sort of research of characterizing the different types of different types of synovial inflammation in patients with rheumatoid arthritis. And we are one of the sites internationally in this huge consortium. And it is not just rheumatoid arthritis. It is actually a number of different autoimmune diseases, of which rheumatoid arthritis is only one. And I show you this to show you, to basically reinforce what a huge undertaking this sort of research is. And the goal here is really to bring precision medicine and new therapies to patients with multiple autoimmune diseases, including rheumatoid arthritis, psoriasis and psoriatic arthritis, Sjogren's, and lupus. And I also just wanted to give you sort of the financial scope of this sort of thing. So it's a huge undertaking. It's $58.5 million over 5 years for all of the different autoimmune diseases. And it's a mixture of medicine of money from the NIH and industry and also some nonprofit funding. And if you would like to learn more about this and learn really more about what each of the parts of the Accelerating Medicines Partnership Autoimmune and Immune-Mediated Disease Program is doing, it is through this website, which is the NIAMS, NIAMS at the NIH. So in the last few minutes, I want to spend some time thinking about where we are in rheumatoid arthritis and what the future holds. So If we think of precision medicine and sort of what it takes to get there as like a linear journey, we have to start by studying the disease-affected tissue. In rheumatoid arthritis, we've sort of only been doing this recently. I don't know if you noticed, but some of those initial papers I showed you about the different subtypes of rheumatoid arthritis were as recent as 2018, 2019. It's actually in terms of like the amount of time it takes to do research and really turn research into changes in patient care that is relatively recently. And so we have really started to study the disease-affected tissue in a real way in rheumatoid arthritis. And I think it hasn't been super easy because it's not something that physicians or researchers really thought a lot about until recently. And so we've been working really hard to understand what types of cells are present, what pathways are activated. Is this different in patients who have never been on a medication before versus patients who have been on three different medications before and are going on to their fourth? Those are the sorts of questions that we've been working really hard to understand. We haven't totally gotten there, but I think we have a pretty good handle on things. And in each of those subtypes of rheumatoid arthritis synovium, there are research groups who are working really hard to understand specific cell types or specific molecular pathways to see if we could eventually get to this end goal, the developing of new treatments. So although I've written this as like a linear line, it doesn't quite stay that way. In here, we've also, in the next sort of stage, is to relate tissue differences between patients to treatment response. This is crucial for understanding, especially in a disease like rheumatoid arthritis, where we have medications, right? It's not that we don't have medications to treat our patients with. We have good medications. We aren't always the best at figuring out the best one for the best patient at the right time, right? And so we are now doing a lot of work relating tissue differences to treatment response. And again, this isn't really just like a one-way street. We are then going back as we find out that, oh, wait a minute, there's a group of patients that don't respond very well to treatment Z who actually have this finding in their synovium. And then we go back and we look at the disease tissue again for all that and say, like, what's happening with this one factor? And that sort of ends up being like a sort of an iterative cycle as we go back and try to really understand the disease well. So the next, after we have related disease, the tissue differences to treatment response, then we have to use the tissue data to robustly predict treatment response. When I say robustly here, I mean we need a test that a doctor could order for a patient and say, if the result is this, we do this treatment. If the result is that, we don't do that treatment, right? And that's what we really haven't gotten to yet. So I think in rheumatoid arthritis, we're kind of here. We have related tissue differences between patients and treatment response, but we haven't really gotten to the point where we can make specific recommendations for specific patients about what treatment is really going to be the best for them based on specific molecular patterns in their tissue. And of course, sort of the holy grail is developing new treatments. I think we are all very hopeful that we will find something in this huge, long process of trying to better understand the disease and how it relates to treatment. We hope we will find something that will let us develop a novel medication or therapy or some combination that will really help us better treat rheumatoid arthritis. Maybe it'll be a more exact sort of treatment so that there's less sort of general side effects like the general dampening of the immune system that is a side effect of many of our medications. We also hope to be able to come up with therapies that last longer. Many patients have the experience that they're doing well in medications. They'd like to be able to stop or taper off their medications. And for some patients, that works. But for other patients, they have a flare, they have high disease activity after trying to come off medications. Right now, rheumatoid arthritis is really a pretty lifelong disease. Not for everybody, but for many patients, it's lifelong, and we don't have any way to really have a cure. That, of course, is like really the gold star goal over everything, but we aren't there yet. So we have this whole pathway and lots of science and other research to do in order to get there. So with that, I'm going to make sure we have plenty of time with questions, but I just wanted to end by thanking you guys for being here. Every patient who participates in and also engages with research really helps advance precision medicine for the future of RA. We cannot do this research without our patients, and we cannot do this research without funding from both the government, foundations, and many, many sources. So with that, I will see